Hi everyone. As yesterday, we're going to wait for a few minutes to see if the participant number is going to stabilize a bit, because right now it's still going up. And then in 2 minutes, we're going to start. Or maybe in 1. I think I would just start with the general part right now. Hello and welcome everybody to the second day of the 4th Transatlantic ECI GPCR Symposium. I think we had a blast yesterday, and I hope that we have one today again. Today we have 3 sessions with short talks, starting right away with session 3 on cell biology and physiology of GPCRs, followed by session 4 on innovative methods and technologies. And after that, just as yesterday, we're going to go into the lunch dinner sessions and the poster session one after the other. The lunch dinner session today is going to be a little bit different because you have the opportunity to talk to all of the short talk speakers again, who are going to be in rooms, breakout rooms, that correspond to the session that they gave their talk in. And then we will have one on communication and one on GPCRs in industry. After the poster session, we're going to continue with session 5 with short talks on computational approaches and structures in GPCR research 2. This is a continuing theme. We had one of those yesterday already. And afterwards, it gets really interesting because we have a short break for award voting. Ian is going to say something about that then. And we're going to finish the day off strong with our second keynote lecture by Ashish and handing out of the awards virtually and closing the symposium. So I'm really excited. And we're going to start right away with session 3 on cell biology and physiology of GPCRs with 4 speakers, Catherine, Annabelle, Edda, and Silvia. And I think Edda is going to start off with her talk on GPCR helix-bundle and C-terminus mediated distinct beta-arrestin conformational changes and functions. I'm going to stop sharing the screen, and I'm going to give the floor to Edda Matthees to start her talk. If she wants to, but she does. You just need to unmute yourself.
Yes, okay, sorry about that. I hope that you can see my slides.
We can. One thing that I forgot and that I need to mention is there's, just like yesterday, some organizational remarks in the chat and also the email address that you can contact if you have any problems. If there's technical problems, you can see Tony and Louise, just as yesterday, who are going to help us with technical support. Thank you for that. So you can also message them if there's a problem. And I take it away.
Okay, thank you so much, Judith, for the kind introduction. I'm very happy and thankful to the organizers for giving me the opportunity to be presenting this work today. So we want to talk about how GPCR domains generally dictate beta-arrestin conformational changes and functions. The great thing about the symposium is that I don't have to introduce GPCRs to you or that they are important drug targets. So this is why it's interesting for us to really understand how the regulation of these receptors is working. And this is what we were focusing on in this project as well. So generally, we just distinguished very roughly between the transmembrane helix bundle and the C-terminus of GPCRs and wanted to see which aspects of receptor regulation are actually dependent on the transmembrane helix bundle or more dependent on the C-terminus, or which of them actually need an individual combination of these two domains. To work on this question, we focused on two very famous receptors in the field, which is the beta-2 adrenergic receptor on the left side, color-coded with this orangey yellow color, and the vasopressin 2 receptor, which is a class B receptor with respect to β-arrestin interaction. So the vasopressin 2 receptor is known to very strongly interact with beta-arrestins and also co-internalize with them. And the beta-2 adrenergic receptor, just as a reminder, is a class A receptor with respect to beta-arrestin coupling. So it shows a very transient interaction. So this was a very interesting finding, basically, already, which is known in the field. And we wanted to check how the receptor domains actually influence this. So to work on this, we decided to use receptor chimeras, where we used the transmembrane helix bundle of one receptor, combined it with the C-terminus of the other receptor and vice versa. So we have the V2 beta-2 receptor and the beta-2 V2 receptor sharing the same C-terminus as the vasopressin 2 receptor, also seen in the color code indicated here in blue. So we actually looked at a couple of different things. We looked at receptor phosphorylation with phosphosite-specific antibodies and also checked the beta-arrestin recruitment, but I will not focus on these things today, just due to time reasons. We also measured beta-arrestin conformational changes for all of these receptors when interacting with beta-arrestin, and also measured receptor internalization and beta-arrestin co-internalization, as this is an important factor to determine this class A, class B terms for these receptors. So this is actually what we're focusing on. And we also measured this in different kinase-dependent conditions, as we know that the GRKs, the ubiquitously expressed ones are GRK2, 3, 5, and 6. Those are the important mediators of the arrestin interaction with these GPCRs. So we have our GRK2, 3, 5, 6 knockout cell line, which we call delta Q for the quadruple knockout. We have the endogenous GRK expression in our control cells, which are HEC293 cells. And then in the knockout cell line, we also checked for GRK2 overexpression or GRK6 overexpression to see individual kinase effects. We also studied ECG phosphorylation over time, but this I will just shortly show you in the end. There's more information on my poster, so feel free to pop by later. So we measure beta-arrestin conformational changes. In this case, I'm focusing on beta-arrestin 2 by using our intramolecular nanobread sensors, which were published in 2022. So we can individually label a specific site in the beta-arrestin and then measure the conformational rearrangements upon interacting with the receptor, so upon receptor stimulation, as a change in the bread signal between the donor, the nanoluciferase at the beta-arrestin C-terminus, and the individually labeled sites. So you can imagine that this is quite a lot of data, and you can see here just one example I wanted to point out, because if you focus only on the very last point, this would be a very strong false positive, especially for the range of bread changes we see for the beta-2 adrenergic receptor. But we measured concentration response curves for all of these conditions so that we are able to actually investigate the hill slope of this bread changes that we measured, and also the EC50 of this, so that we can distinguish between noise, basically, because you can see that this is basically just roaming around zero, and the nice signals that we want to get, so that we can distinguish between responder positions and non-responder positions, so that we not take into consideration, for example, this 10% of bread change at this position for the beta-2, because this does not seem to be pharmacologically relevant. So here you can see just the highest stimulating concentration for the beta-2 adrenergic receptor. We used ISO, and you can see that we were able to measure concentration-dependent responses here. And you can see that here the values in the C domain of the beta-arrestin are 0, which does not mean that we did not measure this. It's just that these were not concentration-dependent responses, so we classified these as non-responders based on the Hill slope and the EC50. For the vasopressin 2 receptor, you can see that we were able to measure concentration-dependent changes also in the C domain of the β-arrestin 2. So we wanted to check how this looks like for the 2 chimeric receptors. And in blue, you can see the endogenous kinase expression always. And you can see that, for example, for the V2 beta-2 receptor, we were able to measure flash 10 conformational changes in the β-arrestin 2, but not for the beta-2 V2 sharing the same C-terminus with the vasopressin 2 receptor. So you can see that the conformational rearrangements in the beta-arrestin seem to be differentially dependent on specific receptor domains. Actually, we also measured all of this in presence of GRK2 or 6. As I mentioned, I don't want to go into too much detail here. I just want to point out 2 things. So especially for the receptor chimeras, you can see that the overexpression of kinases drastically boosted the dynamic range that we were able to measure. And another thing is that you can see that we found, especially for the vasopressin 2 receptor C-terminus, strong differences in the changes that we were able to measure in β-arrestin 2, dependent on whether the GRK2 or the GRK6 is overexpressed. But let's focus on the upper panels just for time reasons as well. So we wanted to check which domains are actually important for the transferability of these conformational changes, right? So to basically compare the patterns that each receptor induced in the β-arrestin 2, we normalized to each respective maximum that you can see here highlighted in green. We wanted to compare the wild-type receptors. I just show it for one example, which is the flash 4 position in the N domain of β-arrestin 2 here. Because you can see that with the wild-type beta-2 receptor, we were not able to measure any concentration-dependent conformational changes at this position, whereas with the vasopressin 2 receptor, we've had a strong response. So we wanted to compare whether the wild-type receptors are already different to basically get, in the end, maybe one value, which we then call the transferability coefficient for each position. So here for the F4 example, you can see that there's a strong difference between the 2 wild-type receptors. So if this position is tail transferable, so dependent on the receptor C-terminus, then the different cores should be quite different from each other. So we compared the wild-type receptors with the chimeric receptors sharing the same core, and then got a value for this flash 4 position in control cells of around 2. So it's a strong positive value, so indicating that actually the C-terminus plays an important role in the extent of the conformational changes measured at this position. So if that is true, the transferability value for the different cores, if we compare these, should be quite low, which is the case for the flash 4 position here. And then to basically combine these values into one, as I said, to get a transferability coefficient for the individual positions, we just subtract the transferability core from the transferability tail value. So if we have a positive value, that means that the C-terminus seems to be more important in mediating this conformational rearrangements than the transmembrane helix bundle, short the core here. However, if we have a negative value, then this means that this value is greater, so the transmembrane helix bundle plays a more important role. So for the flash 4, it's quite clear. You can see that we don't have any concentration-dependent signal for the V2 beta-2, and we have one for the beta-2 V2 sharing the same C-terminus as the vasopressin 2 receptor. And indeed, the value also indicates that the transferability of this position seems to be strongly C-terminus dependent. So we translated that into these bubble plots, also indicating the role of the wild-type receptors, or the difference of the wild-type receptors in the size of the bubbles. And you can nicely see here on the surface projection, indicated by color, that the N domain conformational rearrangements in β-arrestin 2 were more C-terminus transferable, which makes sense because this is also where we know that the phosphorylated C-terminus binds. We also, as I said, investigated receptor internalization and co-internalization with beta-arrestins. Beta-arrestin 2 here specifically, we utilized confocal microscopy to do this, but focused on only one concentration. And just as a short example, you can see here, if we compare the colocalization of beta-arrestin 2 with the RAB5 as an early endosome marker, we can see that this colocalization is actually dependent and transferred by the vasopressin 2 C-terminus, because for the beta-2 adrenergic receptor, as expected, we don't see a significant increase upon receptor stimulation, and also for the V2 beta-2 receptor chimera sharing the same C-terminus as the beta-2 adrenergic receptor, there was no significant increase in beta-arrestin colocalization with this early endosomal marker. So this fit quite well with the previous literature. As I mentioned, we also investigated the time profiles of ERK phosphorylation using Western blot here in the control cell line with endogenous kinase expression and the knockouts. Just as a very short visualization, you can see that the time profile here is more similar for the receptors sharing the same receptor core. So we concluded that the ERK phosphorylation profile over time, how this is influenced over time, is actually dependent on the receptor core. So we wanted to also have a transferability coefficient for these kind of investigations as well. So you can see a similar bubble plot as I've shown you before, but this time focusing on the ERK activity, which means the ERK phosphorylation in this case, and also the receptor internalization and beta-arrestin co-internalization. Using a similar analysis, if you want to have more details on this, please feel free to ask me later in the lunch session or at the poster, of course. So you can see with this analysis that we set up the ERK1-2 profile over time is helix bundle transferable, so more dependent on the receptor core, and the beta-arrestin endosome delivery is more C-terminus dependent. So with this, I'm at the end of my talk, and this is just trying to visually attribute the findings that I've just shown you to the schematic that we have seen in the beginning. I want to thank, of course, the organizers for giving me the opportunity to talk to you. I also want to thank my supervisor, Professor Carsten Hoffmann, and all of the people in the lab that made this project possible, especially also Dr. Raphael Haider, who's now at the University of Nottingham, and our colleagues in the Stefan Schulz lab, Nina Blum, who's working on the phosphorylation-specific antibodies, which I did not have the time to show you. And I'm open for questions now or later anytime. Thank you.
Thank you so much, Edda. Lovely talk. Did we have any questions from the participants? Wait a second to see if anybody wants to put their question in the chat or raise their hand. If no questions from the audience, I'll ask one quick one. So we saw that you could use these transferability ratings for both the flash probes and for the signaling outputs. And do those correlate? Can you correlate specific flash probe dynamic changes in arrestin structure with the signaling outcomes also?
That's basically the holy grail, right? If we can connect these conformational rearrangement with actual functions. In fact, there seem to be some things that meshes as also positions in the C domain of beta-arrestin have been reported to be involved, for example, in beta-arrestin, in modulation of ERK signaling in earlier studies. And this seemed to fit quite well, as the one position that was transferable with the receptor core was a flash 10 position in the C domain of beta-arrestin, and also the time-dependent ERK profile was core dependent. So at least it was not contradictory, but I think to actually say that this is directly related, we cannot really say based on this. But that would be very interesting to be able to clearly answer yes. Thank you.
Thank you.
Thank you, Edda, again for the talk.
Thank
I
you.
do have a question, but I'm going to ask that during the poster session, I guess.
Great, thanks.
And I would like to introduce the second speaker for today, who's Annabelle Milner. And she's going to talk about lactate-dependent regulation of the HCAR1. And I think she can introduce this topic way better than I can. So the floor's yours.
Thank you, Judith, for that kind introduction, and to the host for inviting me to talk. So like Judith just mentioned, I will be talking about the lactate-dependent regulation of HCAR1 signaling and how this influences the intestinal epithelial barrier. So many of you have probably not even heard of my receptor that I work on. It was de-orphaned in 2009. And since then, only 140 papers have been published characterizing all functions of this receptor. However, what is known is that the lactate-activated HCAR1 plays a role in inhibiting lipolysis, reducing inflammation, and promoting cancer cell growth. And so in humans, there are two forms of lactate, both L and D, which are stereoisomers of each other, and they are metabolized and produced by very different pathways. So L-lactate is predominantly formed from anaerobic respiration, whereas D-lactate is primarily formed from gut microbiota fermenting dietary carbohydrates. L-lactate can also be produced via that latter pathway. That makes the gut one of the only places where both the stereoisomers can be found. And so my project is really interested in understanding how the two isomers of lactate can activate and regulate HCAR1 signaling pathways, some of the regulatory mechanisms behind this, and the physiological effect. So for the purpose of today's talk, I'll be focusing on how lactate acts and regulates intestinal barrier function. So to do this, we started by characterizing the G protein coupling pathway for this receptor using an intestinal epithelial cell line called CACO2s. So CACO2s endogenously express HCAR1, and HCAR1 is known to couple through the G-alpha-I signaling pathway. This means, just as a very brief summary, this means if the receptor is activated, couples to G-alpha-I, we then see a decrease in cyclic AMP levels. So using glow sensor technology, we can express the glow sensor plasmid in the CACO2 cells and measure real-time inhibition of Forskolin-induced cyclic AMP levels. And so I've done different concentrations of L-lactate and D-lactate, and the dotted line along the where my cursor is the maximum Forskolin response. And you can see that all of the ligands tested did inhibit the cyclic AMP levels. And then when we used an inhibitor of the G-alpha-I protein, pertussis toxin, which is shown in grey, we then lose this inhibitory effect. So to quantify this data, we can look at things like the area under the curve and the kinetic profiles using linear regression analysis. And we can see that L-lactate is a lot more potent than D-lactate and also has a faster onset of activation compared to D-lactate. So I mentioned that I was using these CACO2 cells, which are intestinal epithelial cells, and they are derived from colorectal adenocarcinoma. They differentiate over 18 days and form polarized cells, which means that the top and the bottom of the cells are different. The apical side or the top side of these cells form microvilli, and the basal side of the cells are then exposed more to the blood barrier. They also form these kind of characteristic tight junction complex proteins, which brings the cells very close together. And so we can kind of use markers of the microvilli and tight junction protein to get an orientation of the polarized cell. And so hopefully when I play this video here, in the green channel, you will see ezrin, which is a protein expressed in microvilli, very much at the top of the Z stack, whereas the red channel, which is Z01 of the tight junction, is more towards the basolateral side. And then you'll see the kind of membrane that they grow on come in. So this led us to wonder whereabouts HCAR1 was expressed within these polarized cells, and did it make a difference what side the receptor was signaling from? And so unfortunately, there are no antibodies for this receptor, or at least no specific antibodies for this receptor. So to overcome this, we made an N-terminally flag-tagged receptor that was then stably expressed in these CACO2 cells, and then differentiated the cells. Using, again, the Ezrin marker for the microvilli expression for the apical surface of these cells, we found that HCAR1 was then predominantly expressed on the apical surface when we quantified this. So the next step was to work out what HCAR1 was doing in these cells and what could it signal from kind of the plasma membrane, the apical surface of the plasma membrane. So I mentioned these CACO2 cells form tight junctions, and this can be used then for electrophysiological studies to measure cell permeability. And to do this, we have kind of this device here that passes an electrical current between the cells. And this, as the cells form tight junctions, we get a resistance to this current, and that can be measured over time. So then what was really interesting was then when we differentiated these cells and measured that resistance to the current over time, we found that the CACO2 cells that were expressing the flag-tagged HCAR1, so effectively an overexpression system, actually had a lot greater resistance to that current than the endogenous wild-type cell line. So this made us wonder whether HCAR1 could be involved in maintaining and promoting intestinal barrier at all. So the next step was then obviously to investigate this, and we did this by adding L or D lactate to the apical or basal chamber of these CACO2 cells and measuring that difference in kind of the tight junction formation. And you can see when we've added L or D lactate to the apical surface, we see an increase in reading, meaning that the cells are becoming more resistant to that current, and that means that they're becoming tighter to one another. This is then, these changes are then enhanced or exemplified in the overexpression system. When we then added L or D lactate to the basal chamber, we didn't really see much of a change in the readings. And so for the purpose of this talk, I'll focus on the apically added ligands. So not only could L and D lactate change the not only could L and D lactate change kind of the tightness of these cells, we wanted to see if it also changed the cell permeability. And so to do this, we use a fluorescent probe called Physiology that we added to the apical chamber along with L and D lactate. And then you can quantify and measure the amount of fluorescent probe that can pass through the cells. And you can see then from the graph on the right-hand side, when we've added L or D lactate to the endogenous or kind of the overexpression system, we see a reduction in the levels of physiodextran that's passed through these cells. So I've talked a lot about kind of how lactate is affecting these intestinal epithelial cells. Is this process HCAR1 dependent? So unfortunately, as it's being quite a new receptor, or at least not a very well characterized receptor, there are no known antagonists. So instead, we use pertussis toxin to block the G-alpha-I dependent signaling. And in the grey lines here, you can see that when we've added pertussis toxin alongside L and D lactate, we no longer really see that increase in the resistance to the current. And again, this is seen a bit clearer with the overexpression system. So this kind of highlights that this mechanism of L-lactate dependent changes in kind of the leakiness could therefore be HCAR1 dependent. So to summarize, I've kind of shown that L and D lactate from the gut microbiota come and activate HCAR1, which is expressed on the apical surface of the plasma membrane to activate a G-alpha-I dependent signaling pathway, resulting in the tightening of these tight junctions to bring the cells together. And we have some data to show that this may be through inhibition of an NF-kappa beta pathway. So why is this important? Well, the gut is home to many pathogenic bacteria and viruses, and so it has to offer a protective mechanism against things that can cause harm. And so far, a lot of this work has been done in quite a healthy system, and so we were then wondering whether these kind of beneficial effects of lactate addition could rectify or help restore barrier function. And so we took then our CACO2 cell model and added LPS to induce damage to the cells. And you can see here, when we've added the LPS for 24 hours, we see a decrease in the resistance, indicating the cells have become more leaky. Then when we add lactate, we almost see this function restored back to almost basal levels, indicating that the lactate can restore the cell permeability. And so this is really interesting from a pharmaceutical point of view, because lactate could be added as a kind of a dietary supplement to people then with inflamed conditions or IBS as perhaps a preventative or a mechanism through HCOL1 signaling to try and correct some of the damage seen in the gut barrier. And so to conclude, I have presented some data showing that HCOL1 is activated by both L and D lactate with differences in temporal activation profiles. I've also shown that L and D lactate can increase intestinal barrier function by activating HCOL1 through G-alpha-I signaling pathways. And again, that LPS-induced damage to tight junctions can be reduced by lactate addition. And so with that, I would like to thank the Hanulolu lab members that I've been doing my PhD work with, and also the organisers for the opportunity to talk and present my work. And I would be happy to answer any questions.
Thank you so much, Annabelle, for excellent talk on a receptor that I at least wasn't super knowledgeable about. It looks like we maybe have time for one question, and it looks like Ben has raised his hand. So Ben, go ahead and unmute and ask your question.
Yeah, really wonderful talk. I was just curious about your differential effects between basolateral and apical addition of the lactate. Do you know what the B to A versus A to B permeability of lactate itself is? And do you think that it's crossing across your monolayer and that's potentially influencing effect, or does it remain relatively tightly contained within the B to A compartment?
That's a very good question. There are other, so there's something called monocarboxylic acid transporters, MCTs, that can transport lactate in and out of and they are found both on the apical and basal side of these cells. So it is likely that lactate can be transported, but I think it's more likely that the receptor is being activated by kind of microbiota-derived lactate, which is found on the apical side, than necessarily being transported from the blood back into these cells and across to activate the receptor on the apical side.
Thank you.
Thank you for the question, and also thank you for the great talk. I again have a question which I'm going to ask you either later in the lunch session or at your poster, if I see you during the poster session. I'm going to hand over to the third speaker of today. Her name's Catherine Demery, and she's talking again. We had three talks about this yesterday, opioids. In this case, a different angle on them. Take it away if you want to.
Hi, everyone. A special thank you to the organizers for the opportunity to present today. And I'm excited to tell you about fentanyl and xylazine-induced overdose. And that means that I'm going to be talking about the effects that are mediated through two different GI-coupled GPCRs. So you are likely aware that we are in the midst of an opioid epidemic. And in recent years, we've seen a really steep increase in the number of fatal overdoses, and that is entirely driven by synthetic opioids, with 92% of fatal overdoses involving a synthetic opioid such as fentanyl, as shown in the upper right-hand corner of the screen. Fentanyl, among other illicitly abused opioids, activates the mu-opioid receptor, which is a GI-coupled GPCR, and through this receptor, it mediates the clinically beneficial effect of analgesia, as well as euphoria and the dangerous effect of respiratory depression. Recently, this has all been made worse by an increase in the use of Xylazine, a veterinary anesthetic, to cut fentanyl. So if we look at the graph on the left, we have detection count on the y-axis, year on the x-axis. You can see that there's been an increase in xylazine detection in DEA seizures, as well as fatal opioid overdoses. Now, xylazine is a non-selective alpha-2 adrenergic receptor agonist, and this is also a GI-coupled GPCR. And through the alpha-2A and 2C subtypes of alpha-2 adrenergic receptors, xylazine acts as a central nervous system depressant. These are autoreceptors located presynaptically in the central nervous system, and so activation of them will decrease sympathetic output and this will lead to sedation, slow heart rate, and other effects that are postulated to increase the risk of overdose. And indeed, in mice, it has been shown that xylazine potentiates the lethality of fentanyl, but a mechanism for this has not been defined. So herein, we hypothesize that xylazine increases the risk of overdose by worsening fentanyl-induced respiratory depression. And in order to test this hypothesis, CD1 male and female mice were injected intraperitoneally with fentanyl, xylazine, or a combination of fentanyl and xylazine. And drug effects on respiration were measured using whole body plasmography, while other vital signs were measured using pulse oximetry. We're going to begin with discussing respiration. So whole body plasmography is a technique in which an awake, free-moving mouse is placed into a sealed chamber supplied with air. And this allows us to visualize their breathing patterns. So when the mouse breathes in, heat and humidity are removed from the chamber, and this is shown as a trough. When the mouse exhales, the same heat and humidity are returned to the chamber, and this is shown as a peak. And so one single breath is comprised of a volume of air that is inhaled and then exhaled. I would also like to point out here that neurons in the brainstem normally trigger involuntary inhalation, meaning that, thankfully, we don't have to think every time we want to take a breath. However, opioids inhibit these neurons, and that's going to come into play in a couple of slides. The first metric that we chose to look at is breathing rate, as shown in the upper right-hand corner of the screen. And when we take a look at the graph, we have breathing rate on the y-axis, time on the x-axis, and the dotted line represents the point of injection. So in our vehicle group here, we do see a slight decrease in breathing rate over time, and that is because the mice eventually relax after the injection and sometimes even fall asleep in the chambers. When we administer fentanyl, we see a really rapid decrease in breathing rate. Xylazine also decreases breathing rate, and to an even greater extent than fentanyl. And when the two drugs are administered as a combination, we see that the maximum depression of breathing rate is very similar to that induced by xylazine alone. However, breathing rate as a metric doesn't capture the full drug effect on respiration, so I also wanted to take a minute to go through some breathing traces really quickly. So in the vehicle group, we can see that there's really frequent inhalation troughs that look pretty regular. When we look at the breathing trace induced by fentanyl, we can see a clear dysregulation of this pattern. And if we look at xylazine by itself compared to xylazine in combination with fentanyl, we can see that xylazine alone looks similar to the vehicle group. It's just slower, it's more spaced out, but the inhalation troughs are still intact. However, in the fentanyl and xylazine group, this pattern is completely different and dysregulated, despite the fact that at this time point, there's a similar breathing rate between these two groups. The patterns are entirely different. So I did just want to point that out. But overall here, we can conclude that the extent of breathing rate depression is dictated by xylazine when the two drugs are administered in combination, and that this is further reduced compared to that of fentanyl alone. As I mentioned earlier, opioids inhibit neurons in the brainstem that drive inhalation. And so I also wanted to look at inhalation flow, or peak inhalation flow, which is depicted in the upper right-hand corner here. Fentanyl very rapidly reduces inhalation flow, whereas xylazine has a much slower onset of effect. And when the two are combined, we see a rapid and much more drastic decrease in inhalation flow compared to either of the two drugs alone. So this is showing xylazine is really clearly enhancing this effect of fentanyl. And to just highlight that we believe this is occurring in a super additive manner, I chose the 15-minute time point. And here we can see that if we look at the theoretical effect size based on fentanyl and xylazine's effect sizes alone, it's only about half of the actual effect size that is observed with the combination. So here we can conclude that xylazine is exacerbating fentanyl-induced changes in inhalation in what appears to be a super additive manner. So in the past two slides, I've shown you that the combination of fentanyl and xylazine decreases breathing rate and inhalation flow more than that of fentanyl alone. So we next wanted to look at how this would translate to changes in blood oxygen saturation as measured by pulse oximetry, because in the event of an overdose, inadequate blood oxygen will eventually trigger cardiorespiratory arrest. So we see that fentanyl rapidly and for a long duration of time decreases blood oxygen saturation. Xylazine on its own actually had no effect on oxygen saturation as measured with pulse oximetry. And surprisingly, when the two drugs were combined, we actually saw that xylazine did not worsen the maximum hypoxic effect of fentanyl. And again, this was surprising because the combination group had shown a lower breathing rate and a lower inhalation flow. And so I wanted to try to explain why xylazine did not further reduce the hypoxic effect of fentanyl by looking at another parameter. And that other parameter is apneas. So an apnea is a period of time during which the mouse is not breathing in. If we remember that inhalation is a trough, then as you can see for this entire period of time, the mouse is not breathing in. And you may be familiar with sleep apneas in humans, and apneas are just have been shown across multiple species to correlate with lower oxygen saturation. So I have a couple examples for you here, just in the breathing traces. In this fentanyl group, you can see an apnea occurring here. And then in the combination of fentanyl and xylazine group, you can see a prolonged apnea. And while you might think that, okay, this is only about 3 seconds, mice breathe an average of 5 times per second. So even in this little bit of time, this mouse is missing 15 breaths, or missing about 10 breaths, whereas possibly up to 15 here. So when I calculated the number of apneas that occurred in the first 15 minutes post-injection, I found that the vehicle and xylazine groups experienced none, whereas the fentanyl, either alone or in combination with xylazine, produced a really comparable number of apneas. So I wanted to directly correlate this with blood oxygen saturation. And so shown on the left is the same blood oxygen saturation graph that you have already seen. And again, I wanted to correlate this with the frequency of apneas. And so I took the 15-minute time point for blood oxygen saturation and correlated with the number of apneas that occur in the first 15 minutes post-injection when oxygen saturation is being depleted. And we can see here that there's a really clear correlation with the vehicle and xylazine groups, xylazine alone groups showing high oxygen saturation and 0 apneas, whereas all fentanyl containing groups had lower oxygen saturation and a higher number of severe apneas, showing that more frequent apneas correlated with lower blood oxygen saturation. So altogether, takeaway messages from my talk today are that xylazine reduces breathing rate and heart rate more than fentanyl. I did not have time to show you the heart rate data, but it's in the title of my talk, so I felt obligated to include it somewhere. And we also found that when combined, xylazine exacerbates fentanyl-induced effects on inhalation. Despite these things, fentanyl, but not xylazine, decreased blood oxygen saturation and induced apneas. And we found that oxygen saturation loss correlated with the number of apneas rather than breathing rate, as might have been expected. And so if you would like to hear about more doses and a lot more results, including exhalation and heart rate, please feel free to scan the QR code here for the preprint, which has a lot more information than I had time to present today. With that, I would like to acknowledge my PI, Dr. Trainer, my other mentor, Dr. Anand, Sierra Moore, who performed most of the blood oxygen saturation experiments, and finally, Dr. Levitt, whose lab I performed the plasmography experiments in. And with that, I am happy to take any questions.
Thank you so much, Catherine, for a fantastic talk on some more opioid work. We do have one question in the chat that we'll ask. So Detlef asks, interesting observation. Did you look at arterial CO2 levels, as body metabolism might be also differently affected by the combination of both drugs, and CO2 is probably the major regulator of breathing frequency? Does the combination of fentanyl plus xylazine affect cardiac output?
That is a really great, a really great question. And yes, so we also believe that the metabolic changes are probably driving or a potential explanation for the xylazine-induced decrease in breathing rate and heart rate. Unfortunately, we were not able to measure arterial CO2 here, but I do entirely agree that this could very much be due to changes in metabolic rate, which we know that xylazine decreases metabolic rate, and that could lead to a lot of the effects that we observe here. I will also say that xylazine produces really unique effects on exhalation, which I didn't have time to show, which of course we normally exhale CO2, and so there's a lot of further implications for that as well. But yes, we were not able to measure that in this current study. Great question.
Then thank you again for the talk and for answering the question, Catherine. And again, if additional questions pop up, you can ask them during the lunch session later today.
Great, thank you.
For the last talk of this session, we have Silvia Sposini with us, and she's going to talk about lipids and unsaturation and the brain and dopamine. And I'm really excited to hear what you want to tell us. Take it away.
Thank you, Judith, and hi, everyone. So my name is Silvia Sposini, and I'm a postdoctoral fellow at Imperial College London and the Interdisciplinary Institute for Neuroscience in Bordeaux, France. As many of you know, GPCR signaling is highly and tightly regulated by membrane trafficking. In fact, GPCRs sit at the plasma membrane, but once they receive their ligand, they both activate signaling responses and start to be phosphorylated by GRKs, which in turn allow the binding of β-arrestins to the receptor. And through these events, receptors start to be internalized into the cells, into endosomes. And from here, we all know very well that receptors can signal, again, from intracellular compartments, as well as being targeted either back to the plasma membrane through recycling or to degradation through the lysosomal pathway. Bearing this in mind, today I will talk about the dopamine receptor D2. This is a class A GPCR, which is GI coupled, and it internalizes through a β-arrestin and clathrin mediated endocytosis mechanism. The D2R is expressed in the brain, specifically in the striatum, and it regulates functions such as locomotion, learning and memory, attention and sleep. And importantly, it is the main target of antipsychotics. Another important feature for my talk is that the membranes of cells in the brain are highly enriched in poly-unsaturated fatty acids. These are phospholipid populations that increase flexibility of the membrane and help preserving the conformation and the activity of GPCRs. How are these related to the dopamine receptor D2? So it has been observed that individuals with mental disorders also show low levels of omega-3 polyunsaturated fatty acids. And it has been highlighted a causal link between the deficiency in omega-3 polyunsaturated fatty acids in dopamine receptors D2 expressing neurons and motivation deficits. So the question we want to try answer is, what are the molecular mechanisms underlying the relationship between polyunsaturated fatty acids levels and dopamine transmission? Well, considering that my area of expertise in GPCRs is mainly related to membrane trafficking, we want to tackle this question through the lens of membrane trafficking, as I mentioned before, considering its importance in receptor signaling and functions. So the main question I will answer through my talk is, do polyunsaturated fatty acids impact dopamine receptor D2 membrane trafficking? So in collaboration with Isabel Alves and Rim Bakush, we conducted this work where we enrich HEC293 cells in either omega-3 or omega-6 polyunsaturated fatty acids that I will call DHA or DPA throughout my future slides. We treat cells for 24 hours with DHA or DPA, and then check whether the enrichment has been successful. And as you can see here, this is the case. This treatment does not impair or affect cell viability, as well as D2R expression at the plasma membrane. So what we did with these cells is to transfect them with flag-tagged D2R and monitor the internalization of the receptor upon stimulation with a synthetic agonist quinpirole. And as you can see from these images, as well as the quantification here, internalization of the receptor is reduced when cells are enriching DHA or DPA. To test whether this is a mechanism shared by clathrin-mediated endocytosis receptors, such as the transferrin receptor, we looked at its internalization under DHA or DPA enrichment, and transferrin receptor endocytosis was not affected. And to test whether other GPCRs, such as the beta-2 adrenergic receptor and other class A very well-studied GPCR, was affected by DHA and DPA, we looked at internalization of beta-2 upon isoproterenol treatment, and observed no effect on its internalization in PUFA-treated cells. So which are the specific steps of D2R that are affected? To answer this question, we turn to turf microscopy, which allows us to really focus at the plasma membrane of the cells. And the first thing we checked is clustering of the receptors. As you can see through this movie, receptors are nicely clustered upon stimulation with quinpirole. When cells are treated with DHA and DPA, we observe no effect, meaning that the clustering of the receptor was not altered by polyunsaturated fatty acid enrichment. We then looked at the recruitment of a protein that has key roles in endocytosis of D2R, such as β-arrestin 2. So cells expressing SEP-tagged D2R and β-arrestin 2 M cherry were imaged by turf microscopy again. And as you can see through this movie, upon addition of quinpirole, arrestin is nicely recruited at the plasma membrane. And when we measure the fluorescence of arrestin in the receptors cluster, what we observe this time is a reduction of fluorescence in DHA and DPA treated cells, meaning that arrestin is less recruited in this condition rather than in the control one. Can we be even more specific? So to do so, we employed a technique which is called the pulse pH assay, which I've now carried out to answer different biological questions. And if you're interested, you can read the Nature Protocol paper from 2020. And this is an assay that is very powerful to really study and isolate newly endocytosed vesicles in living cells. So the principle is that you express in your cells of interest your receptor of interest tagged with SEP. And then you exchange the extracellular solution between a solution at pH 7.4, which allows you to reveal all the receptor exposed at the plasma membrane and in newly endocytosed vesicles. And then when you exchange the solution to one at pH 5.5, only the receptors that have been newly internalized and so protected from the pH 5.5 solution will retain their fluorescence. And by switching these solutions every 2 seconds, we can obtain 2 sorts of movies from living cells, one from receptors at pH 7.4 and one at 5.5, highlighting receptors at the plasma membrane and in newly internalized vesicles. And from these movies, we can also quantify the fluorescence levels in individual vesicles or clusters. That's what we did for the D2R expressing cells at pH 7.4 as shown in this movie. So upon quinpirole addition, we see nice clusters, and we quantify the fluorescence of each of these clusters in different conditions and confirmed our previous results showing that the number of clusters was not altered, as well as the fluorescence in each of the clusters. We then obtain information from newly internalized vesicles by looking at movies at pH 5.5. As you can see here, upon quinpirole addition, the number of vesicles increases and then goes down once the quinpirole is removed from the medium. And we can nicely quantify this here through this cumulative frequencies graph. And when we compare the three conditions, such as control cells to DHA and DPA-treated cells, we can suddenly see that there is a strong decrease in the internalization or formation of endocytic vesicles when cells are enriched with PUFAS. However, what does not change is the fluorescence in each cluster, meaning that each endocytic vesicle contains the same number of receptors, regardless of enrichment with PUFAS or not. And finally, we also image cells, the channel where β-arrestin 2 is expressed with cherry. And again, when we add quinpirole, there is a nice recruitment of β-arrestin to the membrane. And when we quantify this fluorescence, we see a decrease in polyunsaturated fatty acids enriched cells. Meaning, again, that arrestin is less recruited to receptors, clusters, and internalization vesicles, actually, in this case, than in control cells. So the model we propose is that when membranes are enriched with polyunsaturated fatty acids, there is less internalization of D2R, probably due to lower recruitment of beta-arrestin to the receptor. However, we need to keep in mind that this is the situation, normal situation, let's say, in cells such as neurons, where the dopamine receptor D2 is expressed and show higher level of polyunsaturated fatty acids. And this is the situation that we mimic in HEC cells, where polyunsaturated fatty acids show lower levels. My results highlight important questions that we are trying to answer right now, which is like, is the effect of polyunsaturated fatty acids specific to the D2R, or are other GPCRs affected by these? And is the phosphorylation of D2R by GRKs affected by PUFAS? And probably that's the reason why beta-arrestin is less recruited to these receptors. And finally, what are the downstream effects related to endosomal trafficking, sorting, and signaling induced by polyunsaturated fatty acids? And with this, I thank you all for your attention. And I would like to thank my supervisor here in Bordeaux, David Perret, our collaborators, Rim and Isabelle, as well as Pierre Trifilieff and Thierry Dureau, and Bordeaux Imaging Center, and of course, our funding bodies, Wellcome Trust and ANR. Thanks.
Excellent. Thank you for an outstanding talk. And it looks like we do have a question from Aditya, lipids and GPCR trafficking. Unsurprising, Aditya has a question. So go ahead, Aditya.
Thank you for this really nice talk. I was wondering that is it possible that the receptors that are actually internalizing when you enrich with PUFAS, they use another mechanism for internalization? And on the corollary, if you reduce these lipids, do you think? I mean, have you looked at what happens if you reduce levels of polyunsaturated fatty acids?
So your first comment about the receptor using another mechanism, that's very interesting, and we haven't looked at it. However, with a PPH assay, we looked at all internalized receptors, so regardless the mechanism. But yeah, it could be still interesting to look at other mechanisms. Then your second question, oh yeah, reduce the levels. So that's something we are working on, to reduce the levels of polyunsaturated fatty acids in neurons. But this is quite time-consuming approach, because we need to essentially work on the mothers, on the diet on the mothers, to have a diet deficient in polyunsaturated fatty acids, so that the progeny will have membranes with less polyunsaturated fatty acids. But yeah, that's certainly what we would like to do in the near future. Thanks.
Exciting, thank you.
It does look like we have one other question in the chat, and I think we have time for it. So Giovanna says, great talk, Silvia. Will you have any ideas on how the PUFAs influence the beta-arrestin recruitment from a structural point of view?
Yeah, we are working on that too. And actually, I got some ideas from yesterday's and today's talks about maybe the polyunsaturated fatty acids having an effect on the beta-arrestin rather than the receptor only. But what we know on the receptor is that they change a little bit the structure of the receptor, especially in the C-terminal, in the intracellular domains. And what we think is happening is that they mask some serines that are phosphorylated and then recognized by beta-arrestin. And for this reason, beta-arrestin cannot bind. But it would be really interesting to look also at arrestin conformations in presence of polyunsaturated fatty acids. Thanks for your questions.
Thank you so much, everyone. Thank you to all of our presenters and everybody who asked a question. We want to give everybody just a brief break before the next session. So we're going to take just a couple of minutes